for those of you for whom shell was not one of your first words, quick review on mollusks. What are they? Familiar things: snails, slugs, clams, octopus, squid. What unites them? Well, the name refers to the soft body mollusks. Most of them have shells, very important for the fossil end of things. They've got a nice muscular foot in most of them. They tend to have a feeding structure called a radula. It's this usually long ribbon-like structure, tough protein, good for chewing, rasping things, that sort of stuff. And as kind of a bit of a picture of some of this in action, we've got here standard tadpole snail is a common name for the thing. And on this side we've got, let's see, where are we pointing? Somewhere there. You can see it feeding here. So it's using the radula to scrape algae off the side of the aquarium. It's crawling on its muscular foot, and you can kind of see the shell, a bit of the soft body in the background there. Why would we care about mollusks? The Bible doesn't really say much about them, but a couple of their products, pearls, purple dye, get a fair amount of prominence in there. But more generally, there's a fair amount of human use for them. Popular seafood item, used in ornament, important part of the food chain. A lot of them are very good water filterers, important for environmental structure. No shells, other things can live around shells, things pile up, that sort of thing. Some of them very important as intermediate hosts of parasites. Mollusks also, a number of them have rather high levels of conservation concern, especially freshwater and land species. A lot of them at high risk from global warming. That shell, calcium carbonate, just the thing to fizz if you drop acid on. It doesn't do so well with ocean acidification. Mollusks are extremely diverse, both in terms of numbers of kinds. They're second to the arthropods for major groups of animals. Different forms, in terms of different body plans, they're actually the most diverse group. Going with that, lots of different behaviors. God made a lot of them, so worthwhile understanding some about them, protecting them. The picture here shows Timothy demonstrating they're just fascinating. Right around us here in Tennessee, historically, we had the second highest freshwater mollusk diversity of anywhere in the world, Alabama being what's ahead of Tennessee. Well over 100 species of mussels, probably comparable in that of snails, though they aren't as well worked out. Also a good number of land snails present. And if you go back to the fossils, there's all sorts of things. Even the state fossil is a clam. But, say, historically they had this many. There's been a lot of extinctions. Dam a river, you greatly change the habitat. Pollution, obviously bad for the stuff. Poor land use. All your soil runs off into the river is not only bad for your farm, but bad for the river. Now, putting in a dam, it's useful, easier to get your boat through. You can have hydroelectricity, but a lot of species have suffered. Example of the changes, back in the mid-1800s, mussel shoals just south of Tennessee into Alabama had on the order of 70 species of mussel present, lots of snails as well. And as a shoal, shallow area, the Union forces were not able to get their large boats full of soldiers and guns and things up the river very readily, so it kind of slowed them down. So now, focusing a little more, okay, our theme on genetics and paleobiology, how does that tie in? Again, most of them have hard shells, good for having a fossil record. And then mollusks, there's a good number of them still alive that we can easily get DNA, other molecular data, and use that as a clue to the history. So, going as far back as we can with the fossils, the earliest molluskish thing we have is known as Kimberella, latest Precambrian, about 550 million range. Initial fossils, not such great preservation. They were thought to be kind of jellyfish-like, but they were transformed not by waving a magic wand like Cinderella, but rather by finding better preserved specimens. And a couple of key features. Again, what makes a mollusk? You have these rounded impressions. Looks like a nice big muscular foot like a mollusk. These kind of scratch-like things suggesting an animal munching on stuff with a radula. And so overall, the reconstruction is something like the picture to the left in these of something with a bit of a kind of long mouth structure it could reach out and munch on the algae with, and something organic. It's not preserved as a hard part, but something relatively tough over the back, kind of a precursor to the shell. As we move on to the Cambrian, mineralized skeletons become extremely common as we get into the Cambrian, not just in mollusks, but in other groups as well. But in these earliest Cambrian things, we have all sorts of little shells. Not sure what a bunch of them are, but quite a few of them look at least like they could be mollusks. Pinning down for sure is often quite tricky. Some of these have several individual little hard bits of shell on the surface of an animal. This might be kind of a transition between the evidently all organic shell that we had back on Kimberella to a more mineralized shell. In fact, even living today, we've got one group of mollusks, the chitons, that have multiple plates making up their shell rather than one or two for a snail or clam type of thing. A fairly common generic pattern that we see back in the Cambrian is a kind of cap-shaped shell. And figuring out just what group something like that belongs to is kind of tricky, but it does provide kind of a pretty basic starting point. And then we do see some transitions in the fossil record from a cap shape to other forms. So for example, going from a more cap shape to clam, we have a sequence here. Up at the top, we're starting out relatively coiled. As we go along, coil is decreasing, also not so easy to see in the relatively two-dimensional drawing, but these are getting flatter side to side. And you can kind of see in the drawings the groove that I showed here. This reflects flexibility along the back side, so you can come closer and closer to kind of pulling the two sides together. And in fact, this last one at the bottom, Watsonella, starts out as a larva making shell in two different spots, and then it grows together along this dorsal side. So if you just leave it as protein rather than making shell all the way across the back, you've basically got your first clam, which is right in the same time range as when we see these. Another example of this kind of series from kind of generic cap shape to a specific group is seen in the cephalopods. Goes from a kind of medium tall to rather tall cap shape. And as we get to this, you start adding partitions in the back, sealing off the top part of the shell. It's not used so much by the animal. The key innovation going from this to a true cephalopod is the animal having the ability to manipulate what's going on back in those early chambers. And so you can, there we go, along, well, the side there and over here. Let's see. Okay. Well, briefly appearing and disappearing. Along what's the right side of the shell, you can kind of see there's connection, and in the picture of the modern nautilus down below, the little tubes connecting back. What this does, it allows the animal to manipulate relative proportions of gas and liquid in those chambers, and thus control buoyancy, allowing it to float in the water at a given depth. So that transforms it from something crawling around the bottom to something that can float and swim. As we get out of the Cambrian, further changes go on in the mollusks. The cephalopods, with this ability to swim, become the main large swimming predators. Eventually the fish catch up as well. Mollusks spread into freshwater and land out of the oceans. Now there are some groups that do die out, but overall mollusks have tended to make it through okay for a lot of the major extinctions. And of course within the classes we see then more major groups developing as we go on through here. We've got a cartoon version of the earliest known land snail, for example, bit of a mascot at the site. Going across the big extinction of the Paleozoic to Mesozoic, Dinosaurs get going on land. Meanwhile, in the ocean, there are a couple of groups important. Ammonites are a group of cephalopods. They are very diverse in the Mesozoic oceans. They tend to barely squeak through mass extinctions and then didn't make it through when the dinosaurs died out also. Also, in the bivalves, there are a couple different groups that got to be quite large forms, important in the oceans, and then also die out by the end of the Mesozoic. However, at the same time, we're starting to see things look more modern. Some of our modern groups of snails and clams really diversify during this time, and of course they didn't die out with the dinosaurs, but make it on through. Come on into the Cenozoic. There's pretty high turnover at species level for mollusks as you go along. In fact, Lyell, initially drawing up, trying to recognize units of geologic time, one of his main pieces of evidence for dividing up the Cenozoic was what percent of mollusks look like they're the same as living species, what percent are extinct. And so he came up with different groups, and then of course we've divided further since then. These changes in the mollusks can be matched up well to a lot of global changes. So it's not just telling us about mollusks, but other things going on. Major events happening. Antarctica is isolated as South America and Australia move away from it. Change in the ocean circulation. Likewise, cutting things off at Panama, at Suez. Major changes in ocean circulation. And corresponding things living in the ocean also affects the land stuff, how they can move around, things like that. And as we go through things over time, we're seeing increasingly modern-looking things. Not too surprising, but that is indeed the pattern there. Now, how about the genetic part of our symposium title? Well, we don't have as much data on mollusks as I would like, at any rate. Mollusks, although there are a number used as lab animals, they don't have the prominence of, say, fruit flies or things of that sort. And so we're just now beginning to get some relatively complete genomes for mollusks. So there's a lot we don't know yet, but still, we've got some good data on a smaller scale, and these patterns do fit in well with what we see from other evidence. As we get higher level of classification, how do mollusks relate to other things, we're looking a lot at certain anatomical and early development features as well. So, going fairly high level, how do animals relate to non-animals? Well, it turns out, mainly based on molecular similarities, but there are some cell structures that suggest that animals are relatively closely related to fungi, to slime molds, to certain amoeba types of things. Within the animals, mollusks have a reasonable amount of complexity to their structure. So, unlike, say, a jellyfish or sponge, they've got some basic similarities. 3 cell layers, basic to development, left-right symmetry, DNA features, things of that sort. Narrowing down further, important developmental features, mollusks fall into the large group of the protostomes. Most invertebrates are here. As the cells are just beginning to divide after fertilization, you see different patterns in the protostomes versus the deuterostomes like ourselves. Among other things, this means you don't get identical twins by splitting an embryo, you get half an embryo. Within those, the molting things, such as the arthropods, are one quite distinct group. Mollusks are not in that group, but instead are part of kind of a twofold division. On the one hand, there's a number of generally small and obscure things. Probably the most familiar out of them are the flatworms. Larvae kind of similar looking between some of the flatworms and some of these others. But mollusks fall into a group known as the Lophotrochozoans, rather awful looking word, but this reflects similarity in the formation of the body cavity early in development. A lot of them do have some sort of hard shell, though not all of them. And there are a number, they're not the mollusks, that have a specialized feeding structure, a lophophore. It's a structure that's got a bunch of tentacles on it, generally kind of horseshoe shaped. And once again, the DNA is really one of the main things that drives recognizing this as a group. Now, within that group, exactly what's closest to a mollusk is not well resolved. And also, within the mollusks are snails closer to clams, to cephalopods, things like that. Again, we don't have the greatest resolution there. Now, for the fossil end, well, ancestor, probably pretty generic looking, and so it's hard to say, okay, here's a worm-like fossil. Well, that could belong to just about any phylum. So trying to pin down something that's generic enough to be the ancestor, yet recognize how it fits in, is kind of challenging. Another problem, it looks like we had fairly rapid development of things around the beginning of the Cambrian, and so given that, there's not a whole lot of time for features to appear that we can then go back and look at and say, okay, these are united in this. We've got picture here, long branch attraction is a technical term here, but you can see the number of things that would happen in those little red bits that would make A and B similar are going to be relatively few compared to the differences that develop once the two lineages have split. There's even more of a problem in DNA in that, given you've got basically A, G, C, and T to choose from, mutation may go back to some other feature and confuse the picture further. But there is a lot that we can figure out. One question I looked at back with graduate school, are the true oysters closer to pearl oysters or to scallops? There are some morphological similarities going both ways. And so which way does it work out? Well, turns out that both the paleontology and the genetics help with this. Turns out, genetically, very evident similarities between the oysters and the pearl oysters. And on the fossil record, it turns out that the very first oysters were, in fact, still pearly on the inside. By that time, the scallops had already lost the pearly inside. So both fossil and DNA converging on the answer to this long-standing question. People have been wondering since the late 1800s about it. We do see problems. Historically, there's been a lot of, I study the details of the gills, and here's a classification based solely on the details of the gills. And somebody else looks at the stomach and so on. And so, not surprisingly, sometimes one feature may have unexpected issues. This is a tree, you can't read the names, but colors indicate things that are supposed to be the same genus of freshwater mussel. And you can see colors tend to be clumped, but it's far from perfect. And these classifications largely based on one or two features. Yeah, sometimes that particular feature doesn't work. So overall, mollusks are fascinating, fairly important, sometimes imperiled part of creation. Got pictures of some extinct ones from Tennessee down here in the museum collection. We do have a lot of data, but also quite a lot still to do in terms of understanding just how God's made our present diversity and how to take care of it. So, thank you.
So we have time for questions. Any questions from the audience?
What is your personal research right now? What are you going to do next week?
A couple of things. One, with having just recently started Gardner Webb, I'm in the process of trying to get set up to do some of the DNA work. And once I get it up and running, I've got a couple of groups of freshwater mollusks we're trying to figure out what's going on by getting some DNA evidence on it. So that's one thing I'm working on. Another is looking primarily at various fossils, just, okay, what kinds were here? Nobody's actually really looked and seen just what species are present. So see, okay, what was there? What does this tell us about the environment? Things of that sort. So those are a couple of the major areas that I'm working on right now. They're fairly divergent from most of the other mollusks, which was kind of suspected given the difference anatomically, although there's one rather obscure group of, again, kind of cap-shaped shells. All the living ones are very deep sea, and those seem genetically to be a little closer to the chitons. There's also a couple of worm-like, no more than little bits of pieces for shell forms that seem a little closer to the chitin. So they're a very different group from the standard snail, clam, octopus line of things, and they do provide some useful insights that way.
Where is people helping out freshwater lakes?
Well, part of it is that they belong in fresh to brackish water lakes of Central Asia to southeastern Europe and not here. There's a similar, in terms of ecology form, though quite different group of bivalves native to South Asia that's doing the same major negative impact in South America now. And the key issue in both cases is the fact that they have a different life habit from any of our native ones. They make those abyssal threads glue onto surfaces, and so are very good at covering things, whether it's, say, your intake pipe that you want to keep clean, or covering a native mussel and both weighing it down and competing and potentially starving it. So not having had anything that lived quite like that here, there's not much of a way to protect against it and respond. There are a couple of different groups. The conchs actually are the standard, like big pink conch, Florida, Caribbean, are actually grazers but have well-developed eyes. And some of the predatory ones, especially some of the swimming forms, have very well-developed eyes. And then, of course, cephalopods, octopus, squid, are also very good eyes. Well, eye has a pretty obvious function, gather light. And quite a few mollusks have very basic, okay, light versus dark, shadow comes over, maybe you better hide type of eyes. Various ones that are either more active or more exposed have better developed eye structures, and again, well, basic physics, get good use of light, get some lenses in there, things like that. So it doesn't seem too surprising that you'd have some convergent development of similar well-developed eyes in anything that had a need for it. The one that's really puzzling on the well-developed eyes is some of the box jellies, the really highly dangerous sting ones that have extremely complex eyes and no brain to make use of the information. I mean, clams, well, they don't exactly have a brain, but at least they've got nerve lumps, ganglia, that they might do something with it. There's been a lot of looking, given you've got a pretty good fossil record for the mollusks, to try to see what factors may correlate or not as well. And some things seem to make a bit of a difference, like do you live on the surface or buried underneath? Some issues related to size. Do you have lots of fancy features that might be hard to maintain under difficult situations? Are you widespread or local? So some of these have come out with correlations. Some of them, well, maybe mass extinction is just a big catastrophe and it's kind of largely a matter of did you manage to live somewhere other than where the asteroid crashed on top of you? So some of these have pointed to factors that might be playing a role. Of course, also there's a question of year in, year out, what tends to survive longer versus in the middle of a catastrophe. And there's some differences there. Some of the things that you'd expect to seem to help year in, year out, but don't necessarily help you as much when it comes to a catastrophe where everybody's in trouble. Those are much more complicated. I guess you can kind of advanced, use that as a term for them. They represent good, well-developed snails that have gone back to a relatively cap-shaped shell. In fact, there's at least about half a dozen different groups of snails where you see a cap-shaped shell reappearing, kind of simplifying from the coiled shell that's normal for snails. So it's here and there throughout snails, plus you've got this one living deep-sea group, and so then we've got these fossils, and okay, here's a cap-shaped shell. What is it? Unless you've got really good muscle scar impressions or something like that, often it's kind of a, well, we don't really know for sure on some of the, especially the early fossils where you don't have a close modern relative that we can look at the anatomy.
Thank you.